Okay, folks, welcome back. This is again the course in the bioethics program. We have one last item here to cover actually in it's kind of transitional, the beginning of human life before moving full-fledged into the end of human life bioethics. Anyway, at the very beginning, I always like to begin with a little prayer. In the name of the Father, the Son, and the Holy Spirit. O Lord, send forth your Spirit, and we shall be recreated. You shall renew the face of the earth. Amen. Father, Son, Holy Spirit. Okay, any questions or comments before I continue forward? Hearing none.
I have a few, just one question, Father Shofi.
Yes, go ahead.
Because I didn't hear well the last part of the last lecture, the lecture number 4, I believe it was cancer with the pregnancy. Just the
Yes.
ethical issue of the, just if you can tell me just a very summarized issue of this situation, that cancer with the pregnancy.
Right, so this was one of the 2 examples with the principle of double effect that I was trying to illustrate, all right? When we have simultaneously 2 effects from the same single action. First, we have to understand that bioethically, an action is a whole set of individual actions as they were, right? For example, let's say we're doing an ethical evaluation of a surgery, for example, non-therapeutic sterilization, all right? Obviously, that surgery has several different steps, physically speaking, physiologically speaking, all the way from the time that the patient agrees to the surgery or requests the surgery, the signing of all the documents, the anesthesia that the patient takes and all that is pre-op, technically speaking. Then there's the actual intervention and even afterward, the post-op. So you can see that there are many, many different actions involved in the one surgery that we're calling as a whole non-therapeutic sterilization, okay? So bioethically, that whole cluster is considered a single action, as it were, for the purpose of, so we're analyzing the entire event, all right? So applying that, concept to a woman who is pregnant and has also cancer. And so what do we have? We have her life, the life of the unborn, and typically the cancer treatment, which tends to be aggressive, right? There may be actual surgery involved, there may be chemotherapy, radiotherapy, particularly the chemotherapy, and even the surgery itself may be harmful, lethal to the unborn. And so we have the 2 effects of that one action of the therapy, of the healing, the one action. The 2 simultaneous effects would be, the good one is obviously trying to save the life of the mother. The bad one is that in doing so, it is foreseen that her unborn child will suffer damage and maybe even die in the process. Okay, so we have these 2 simultaneous actions. What to do? The principle of double effect applies in this case, as long as we don't target the human embryo directly. See, if we don't target the human embryo directly, we may go forth because the woman has a pathology that is lethal, and she has an ethical right to a stewardship, right, to care for her own body. And there is treatment available, there is protocol available, It's not going to be 100%, but there is some degree of assurance that following this protocol, based on previous cases, she has a chance of surviving the cancer. All right. And so we go ahead and we look at the 5 conditions for the principle of double effect. If the 5 apply, she may go ahead with the treatment in good conscience. And in fact, it was the case of St. Joanna Mola, right? She had developed a cancer. In her case, she decided to forego the treatment. That's what we call heroic. But she may have also chosen ethically the other option, which would have been treatment. And in the treatment, it is foreseen that her unborn child would die or be severely handicapped from the radiation, for example. And it's foreseen, but the unborn was not targeted by himself or herself. It was not essential to the treatment itself, you see. So does that clarify, or do I need to go further? Dr. Farid?
It's very clear, Father Shofi. It was very clear explanation. I'm
okay. So you see, double effect applies in this case. And I say a cancerous uterus, but actually the cancer could be anywhere in her body, really. It's just exactly. With the cancerous uterus that is pregnant, the most dramatic thing is that the uterus needs to be excised because that's where the pathology is, right?
Exactly.
And so she qualifies for a hysterectomy, and the unborn would definitely die. Of course, we are assuming that it's before viability. If it's after 21, 22 weeks, then they may attempt to save the unborn, right?
C-section.
With a C-section and the NICU, neonatal ICU. But before viability, right? That's why viability is also a moving target in the sense that it keeps becoming earlier and earlier as science progresses and this technology progresses. All those instruments are tiny miniature little needles and things that are used for the fetus, right? Anyway, that's the point. And so it's kind of similar to the tubal pregnancy.
Yeah, to the ectopic pregnancy.
Ectopic pregnancy.
Ectopic pregnancy,
right?
yes.
Ectopic pregnancy, that is, as long as it doesn't target the human unborn by himself or herself, then it's okay. You know, it's sad, tragic, but at least there's a justification. Yeah.
Pleasure.
Okay. Right. All right. So moving forward then, today I just want to look at stem cell research and human cloning. They're a little associated, at least technology-wise, and sometimes also in the use. And I will say from the start that this area of bioethical controversy has moved forward a bit, thanks be to God, because of the success, and I'll just say the success of the various adult stem cells that are available today, all right? The various sources of adult stem cells that are available. And so, and they are showing clinically great success. And so that's something positive that has happened, has developed just in the past, I would say 5 to 10 years. Excuse me. All right, so let's go forward then. We're going to look at stem cell research. And as always, we have to make a number of distinctions here that are bioethically relevant, right? The distinction between human and non-human research. Then the distinction between adult and embryonic stem cells. And then we're going to look at other sources of stem cells like cord blood, the induced pluripotent stem cells, the IPS stem cells. And then This regards cloning then, which technically it is a somatic cell nuclear transfer, or just nuclear transfer. It's the technical term for cloning. And the two types of cloning according to their purpose or the intention, reproductive or therapeutic cloning. So let's go right into it. First, when we talk about stem cell research, we need to make two critical distinctions. The first level between human and non-human, and then the second level between adult and embryonic. And I always like to ask the question here at the beginning, if I were just to ask a general question, is the Catholic Church in favor or against stem cell research? Just stated like that, is the Catholic Church in favor or against stem cell research? Now, I can't see or your hands. This is one of the disadvantages of having virtual, is that we can't see everybody as if you were in the classroom, but how many of you would say in favor, how many against, and then how many would actually ask the question, in what sense, right? In what sense? Because the way it's stated, many people think, oh no, the Catholic Church is against stem cell research. But actually, just the way it's stated, I can tell you that the Catholic Church is not only not against stem cell research, the Catholic Church is actually involved in stem cell research, okay? So when I was preparing to get into graduate school for the second time, when I went for my doctorate in genetics there at Purdue, it was the year 2001, and I spent a year in Boston in preparation. And during that time, part of the thing, I took some graduate courses in biology to get kind of updated, but also I did some volunteer research at St. Elizabeth Medical Center. I don't know if any of you are familiar with Boston, but it's a big hub of education and research, right? That's where Harvard is, MIT, Boston University, Boston College. I think it's the highest concentration of research and education, upper level education in the US. At any rate, St. Elizabeth Medical Center is the Catholic hospital of the Archdiocese of Boston. And it is also the teaching hospital for Tufts University. That's another one that's there. The medical school, I think it's called the Kennedy Medical School, which is near downtown Boston. That is their teaching hospital, okay? So they have a whole building that is on the side of the hospital itself that is all research. And they do the labs also, by the way, they're in that building. They do the labs for the hospital itself. And I was doing voluntary research there. And I was not involved in it directly. I was involved mostly in hematology, but they were doing stem cell research in that building. And it's a Catholic hospital associated with, like I say, Tufts Medical School. It turns out that, of course, and I was curious when I heard about it, what kind of research. Then one day well, I'll save that other anecdote for when we go a little further. But then digging a little bit into it, just asking questions, what kind of stem cell research they're doing, it turns out that they were doing adult stem cell research, okay? Adult stem cell research, which makes a big difference. It was not embryonic stem cell research. And you'll see, and it was not human embryonic stem cell research, which is the challenge, ethically speaking. So the first level of distinction we need to make, obviously, is between human and non-human research, in the sense that we may justify some level of non-human research for precisely the sake of medicine, and in the hope that that kind of research eventually will translate into some kind of cure, right, or clinical trials. Obviously, we or the scientific community experiments with animals before they experiment with humans. And typically the lab animal that gets it is the mouse, which are raised in the lab itself and so forth. By the way, at that building, they had a basement, and that basement was all for raising the mice that they used in the research. And I can tell you that the protocol for maintaining those mice is very rigorous and detailed, and it's a manual that is pretty thick on how to properly, humanely maintain those animals because they have to be treated humanely, even though at the end of the day, they may be sacrificed in a particular experiment. Because there is no alternative, right? We need a mammalian model before we go to human. And so that's how it's done. But the sacrificing of a mouse in a lab today has to be justified ethically, not just from the physiological perspective of the actual experimental design, but also ethically. So if there's an alternative to sacrificing the mouse, then that route should be taken first, okay? All right, so that's the first level of distinction, of course. Without going into further detail, I think you understand intuitively that there are certain experiments that may be done in non-humans that should not be done in humans, or even when there is consent. So the type of stem cell research that we're talking about here is with human stem cells. Okay, so right away the issues of consent pops up. So the second level of distinction is precisely between embryonic and adult. And this one is the crux of it, bioethically speaking, because to obtain adult stem cells, so we have an increasing, we have a variety of sources of stem cells in our body right now as we speak, and that are being discovered more and more. I'll talk about that in a minute in more detail. But basically, to obtain those stem cells, it doesn't take, is basically no harm, no significant harm to the human body, all right? Basically, what is needed is consent from the donor. And if the donor is a minor, then consent from the parents, as long as there is no significant harm or risk to the child in obtaining these stem cells. But we have a variety of sources of stem cells in our body, including human blood, that is fairly easy to obtain without any significant harm to the person. In contrast to that, for obtaining the embryonic stem cells, the human embryo has to be killed. You see, has to be killed. And that's the critical issue there, that to obtain embryonic stem cells, the human embryo has to be killed. All right. And so let's look at that, how it happens, because it turns out that the stem cells, the embryonic stem cells, come from the inner cell mass. So here we have a macrograph, a microscopic photograph of a blastocyst. This should be very familiar to you by now. We've seen it a number of times. We can see that the trophoblast, right, on the outer layer of cells that forms the placenta, remember? And then here's the inner cell mass. And these are the embryonic stem cells of human when it comes from a human blastocyst, or non-human when it comes from another animal, another mammal, okay? But these are the embryonic stem cells. So in order to obtain these embryonic stem cells, it's obvious that the blastocyst has to be dismembered. Disaggregated is the technical term, right? Disaggregated, the cells, the cell aggregates has to be disaggregated, compromising the blastocyst's integrity, And then the ICM is put in a Petri dish and allowed to grow in culture media that is mimicking the internal environment of the female reproductive tract, which is where the blastocyst, that's the natural environment of the blastocyst, right? And they will be grown there to confluence, etc. And then there's passage, the whole thing about stem cells in the lab. Obviously, they're maintained under sterile conditions at 37 degrees centigrade, etc. So you see, this is the crux of the issue here, bioethically speaking. In order to obtain embryonic stem cells, whether human or not, the blastocyst has to be disaggregated, meaning has to be killed. Okay? And so that's the crux of it. If it were hypothetically, if it were possible to obtain human embryonic stem cells without significantly damaging the blastocyst, the human blastocyst, then we would revisit the issue of its ethics, of ethical use, all right? But think about that a little bit functionally, because you know, every biological entity is in situ and is a particular place in an environment where it is naturally living and surviving. Excuse me. And the natural environment for the human blastocyst, of course, is within the female reproductive tract, specifically on his or her way from the ampulla to the endometrium, right? So that's the natural environment. And it's not that we're going to go into that voyage there, all right, and interfere with that for the purpose of obtaining stem cells, because imagine how elaborate that is, how risky to the human blastocyst, to the mother herself, even the timing. You know, many women don't really know they're pregnant in the first week of pregnancy, okay? And so really, whereas theoretically we can paint a picture there, in practice, it is practically impossible to do that in the natural environment of the human blastocyst, as he or she is traveling, again, from the ampulla to the endometrium, right? Those 5 to 7 day window there. And then extracting him or her from that environment, trying to pluck out 1 or 2 of these cells. That's another thing too. We don't really know if plucking some blastomeres from the ICM is going to damage the ICM or not. It's going to compromise the integrity of the ICM. Yes, it's true that plucking 1 or 2 blastomeres from the morula seems not to affect the outcome, you know, the healthy outcome of the morula, in the sense that we know with pre-implantation genetic diagnosis through the IVF industry, that quality control is done, in fact, and those human embryos who, human morally, who, from which 1 or 2 blastomeres were plucked and sent to the lab and so forth, that morula continued normal progression into blastocyst, implantation, etc., because then 9 months later, the baby was born normal, right? As far as we can tell. But remember that it's a critical difference, because at the morula stage, those cells are still pluripotent, right? Pluripotent, which, by the way, I'm going to cover that in greater detail right now. But at the blastocyst stage, already they're not pluripotent, they're pluripotent. In other words, there's a first differentiation, first level of cell differentiation at the blastocyst. So that there's a trophoblast and there's an ICM. And they have different functions. In other words, there's the same embryo, right? But the trophoblast becomes a placenta, again, whereas the ICM becomes the embryo proper. And so this first level of differentiation here, of cellular differentiation, is not insignificant, all right, even ethically speaking. We may, I don't know, it's just I'm talking intuitively here, we may in fact be able to pluck a blastomere of the trophoblast without significant impact to the overall integrity of the trophoblast. And it's just my intuition that plucking one of these, it's about 2 or 3 dozen cells, maybe more, that make up the trophoblast. By plucking just one, as long as we don't compromise the outer layer, the integrity as a whole, the overall integrity, physical integrity of the blastocyst, okay? But the ICM, you can see, looks very different. Just by the gross anatomy, it looks very different in cellular composition than the trophoblast, okay? And so it may not be the same. And furthermore, we don't really want to find out, right? Because what if we start plucking out ICM cells, blastomeres, just for the heck of it, and see if that ICM continues further development, just normal development, right? And what are we doing? We're experimenting with human embryos, and the first thing that comes up is, how do we get consent from that embryo to do that? And so, obviously, we cannot obtain consent. It could be lethal to the embryo, and we're risking lethality to the embryo for what purpose? It just doesn't justify, it doesn't really meet the double effect criteria, right? So you see, I think you understand the point. The ICM, bottom line, the human ICM needs to be left alone. Needs to be left alone because that tiny mass of cells is the human embryo proper, who has to be respected just like any adult human being or later stage in development, et cetera, et cetera. Okay. The ICM needs to be left alone. All right, so that's the challenge there with human embryonic stem cell research, is that to obtain them, the human embryo has to be killed. And therefore, at least from the principal perspective, where we have to justify the means, not just the end, then it's not acceptable. Okay, as I mentioned, this is an opportunity to review then the stages of cell potency, right? Which globally, there are 4 stages of potency. First, before I go there, I don't know if you have any particular questions or comments up to now. Nothing. All right, good. So let's go forward. All right, cell potency. We begin with what is known as pluripotent, and here again is the added value of the course, a little Greek and Latin. Potency. This also is an opportunity, by the way, to talk philosophically a little bit. Remember that there's like a tripod in bioethics. It's like a tripod of the academics in bioethics, which is the science, of course, the empirical stuff, but also the philosophy and the theology, right? At least when we talk about principal bioethics, we have to include theology, because that ultimately gives us the fuller meaning of our origin, our purpose, and our ultimate end, and so forth, precisely as humans as God's image. So a little bit of the philosophy then. We have these 2 philosophical categories, if you will, potency and actuality. And unfortunately, many people today never got a basic lesson in philosophy. Okay, and so they're not really very clear, at least formally, on the distinction between potency and actuality. And in my opinion, being involved in bioethics and specifically pro-life for close to 50 years now, I find that this inability, or just say ignorance about the distinction between potency and actuality, is the root cause of a lot of misunderstanding in society today about these bioethical issues. including the beginning of life and the end of life and so forth, okay? But very especially the beginning of life. And this is what I mean. So potency and actuality. Let's say we are actual human beings, okay? We're all actual human beings. Biologically, we can say that we belong to the human species. In other words, if we have a human genome, and that is empirically measurable, quantifiable. So look at the elegance of the compatibility between science, philosophy, and theology when we do a proper analysis, all right? So we can start with the facts of nature. That's also why, at least in the Catholic Church, when we do bioethics, we do what is called natural law bioethics. Natural law meaning that it's based on nature. We allow nature to tell us what's happening, the facts. And then we do the bioethical analysis based on those facts, just like I pointed out to you just now. What is the factual existence of the human blastocyst, right? What is in actuality that we have? What is it? The quid est, what is it? All right, so we are actual human beings because we have a human genome with its variations, so that allows for individual particular variation within the species. But the variation is accidental. In other words, in fact, the fact that we have blonde hair or black hair or no hair, is accidental to the fact that we are human. All right. So what is essential is that we are human. That is the substance, what we call substantial in philosophy. And there's even a grammatical consistency with that, which is beautiful. It's excellent because we express ourselves with a grammar, right? We communicate grammatically, and that communication is also can be a logical, reasonable communication. So the equivalent, the grammatical is equivalent to substance or essence is what we call the noun, the common noun. All right, it answers the question, what is it? What is it? What is it in front of me right now? Well, what's in front of me right now is a computer. A computer is the noun, you see, and it's a common noun. There are many different types of computers, right? But the common, and that is accidental. The different types of computers is accidental. But what is substantial about all computers is that they compute, that they compute, all right? And so human, when we say human being, right, or humanity, substantially, we are one humanity. And so that is the noun, the human. Now, potentially, we may be any number of possibilities, all right? For example, potentially, men may be husbands, women may be wives. Some actually fulfill that potential and some don't. But any, let's say, any newborn boy potentially may be a husband, and any newborn girl potentially may be a wife in the future. All right, some will and some won't. But that doesn't change the fact that they're actual human beings with potential for being husband or wife, you see? Now, what cannot happen is that a baby boy can potentially be a wife, because that doesn't fit the natural law definition. By definition, wife is female, all right? And unless we get into ideology that is totally inconsistent with nature, all right? By definition of the word wife is female. And by definition of the word husband is male. So a baby boy potentially can never be a wife. And a baby girl potentially can never be a husband from nature. Mhm. All right, so you see, also to any boy or girl born within the territorial United States, which includes Hawaii, okay, may be president of the United States, because the basic requirements, as I understand it, is that the first basic requirement, let's say, for being an American president is to have been born within the United States. So naturalized citizens don't count. No, don't count, don't qualify. Don't qualify. It's just I don't know if it's constitutional or what, but that's the requirement. All right. So any boy or girl born, any human being born in the United States potentially may be president. That means that anyone not born in the United States does not have the potential to be an American president. They may have the potential to be president in other countries of the world, or kings or queens, I don't know, but not in the US. So this thing of potentiality is extremely important. Now, let me apply those concepts to the human being. And that's precisely why the very first lecture in the beginning of life course is on the biology of fertilization and the entity of the zygote, of the human zygote. Okay, and perhaps people have told me you just spent too much time in detail in that biology, in that first lecture that you give with regards to fertilization and arriving at syngamy, right? Arriving at the zygote with a full human genome. And I say, well, perhaps a sin of excess, but I'm trying to do it because this is a fact of nature that none of us invented, or some understand it, some don't understand it, but that doesn't change the biological fact that when we have the fertilization of a human egg by a human sperm under normal conditions, and that zygote reaches syngamy, we have a human being. We have a human being. We have that first stage of human development. Obviously, that human zygote does not look like you and I right now, or even a newborn baby, or even a 3-month-old fetus, or even a 1-month-old human embryo, right? Doesn't look like, but that's the external. None of us look exactly like each other, right? Even identical twins have very detailed differences in them. So it's not about the external looks at all. That is totally accidental. It's about the genome. It comes down to the genome. If this entity has a human genome and is alive and is functioning, is metabolizing normally, then by definition, that's a human being. And so we can say that the human zygote is a human being, actual human being, the actuality, with potential for further development. You see, so how the potentiality comes in? Potential for further development. And when we apply that concept to the whole of life, from conception to natural death, we see that we are actual human beings who are always in potential for further development until the day we die. And so there's the issue of potential for further development is there as long as we leave that entity, that biological entity, in his or her environment, natural environment, right? Which is within the womb for the first 9 months, and then out in this cruel world for the next 8 decades or whatever the good Lord has us here for. Right? So potentiality is a very important philosophical category that has tremendous meaning and application and clarification in the realm of bioethics. So potent, and you see science uses that terminology also. Potency means capacity for further development, here understood in the anatomical and physiological sense, because we're talking about cell potency. All right, so we apply it and it's consistent. The concept of potency is consistent when we apply it to a physical biological entity like the cell, as you know, the basic unit of life, organic life, right? As far as we know. All right, so pluripotent, then just the prefix will qualify. So here is another example of Latin that it has the adjective and the noun together in a single concept, but we know that the adjective qualifies the noun, right? So sky is the noun. I'm looking at it right now, and I can say blue sky. Actually, the sky that I'm looking at right now is white sky because it's covered with clouds. All right, but white is the adjective, and it qualifies the noun. But it doesn't change the substance. The fact that the sky is white or blue or orange does not change the fact that it's a sky. All right, it's just qualifying an accident of the sky, which is its color. So pluripotent, right, is a cell that has the capacity, has the potential to develop into any cell of the body, to develop or to specialize into any cell of the body. And those cells are the zygote. You can see again here is before syngamy, actually, you see the two pronuclei. I've shown this micrograph before. Let me see, just, okay. To amplify a little bit, hope you can see it. See the two pronuclei fusing. You can see, in fact, we have the whole cumulus here of the corona radiata, zona pellucida, the two polar bodies embedded within the zona, okay, etc. So that's a zygote, single cell, pluripotent, has the potential, and in fact, when left alone, becomes the entire embryo, right? Then also, spoken before, the morula. These are macrographs. Whoops. Okay, let's put it here. Each one of these could be at the 8 or 16, probably at the 16 cell level, right? How they duplicate at every passage, at every mitosis. Each one of these blastomeres is still pluripotent, and talked about that already. So that means that if any one of these is plucked out, this single blastomere has the potential for becoming a whole human embryo. Each one of these blastomeres, yes, and that's another thing. If you think about it, when one of these blastomeres is plucked out and sent to the lab for analysis, now potentially we have the possibility of generating a new human embryo, that is essentially a twin, an identical twin to the rest of the morula that was left behind in the incubator. And if that blastomere is now going to be analyzed like a karyotype and so forth, obviously that blastomere is going to be killed, right? Which could mean the killing of another human being. So you can see the ethical morass that we'll get into with that pre-implantation genetic diagnosis. All right, so anyway, for the purpose of this thing, pluripotent are zygote and morula. Then once we go into the third stage of embryonic development, the blastocyst, because we have two cell layers here, the trophoblast and the ICM, then we can see that there has been cellular differentiation that has occurred already. These are regulatory genes that kick on very early on, right? That do this, they guide this differentiation, the first cellular differentiation between the pluripotent blastomeres and the pluripotent, now they're called pluripotent blastomeres of the blastocyst. This means that, in principle, trophoblast blastomere may not become, is restricted, can only become placenta and will not become part of the embryo itself. Whereas the ICM will become the embryo himself or herself, but would not become the placenta. All right, so there is restrictive teleology, a goal or end here. And therefore it's now pluri, but not totipotent. It's within a range, all right? And the range is that, especially for the ICM, the blastomeres of the ICM potentially may become any one of any cell in the body, but not the placenta. And that moves on to the fourth stage, which is going to be the gastrula, right? After implantation, gastrulation, when the ICM starts elongating and becoming bilateral, and then we get the formation of the three germ layers, ectoderm, mesoderm, and endoderm, right? We know that all of the organs and tissues of the body will come from these three germ layers. They're known as germ layers, ecto, meso, and endoderm, okay? And this is at the gastrula stage. And so there's further cellular differentiation there. And also keep in mind that naturally, differentiation is a one-way street, is directional in that sense, is one way toward further and further specialization up until full specialization, fully specialized cells, which are typically the cells in our body, fully specialized cells. So the gastrula is definitely more specialized than the blastocyst, okay? Specifically with the three germ layers from which all of the organs and tissues of the body will come. But generally they're known as a pluripotent. In other words, let's say the cells of the ectoderm may become any of the organs and tissues of the ectoderm. For example, the two typical ones are skin and also the nervous system. The nervous system, interestingly, comes from the ectoderm. All right, I'm not going to go into details there. You can look it up. So we can see that there's a gradual restriction as cellular differentiation progresses in time and in situ, in place. Finally, we get to, well, actually the third stage is called multipotent. Multipotent is the different organs and systems of our body. Whoops. What happens in multipotent, here's just the circulatory system, probably respiratory system, cardiac functioning. Basically, multipotent is just the cells in our various systems, the half dozen, 2 dozen systems that we have, depending on how you classify them, that have the potential for becoming any cell within that system itself, but it's system specific, all right? So basically at the level of pluripotent is broader, is basically within the 3 germ layers. But once we go into the fetal stage after the 2nd month, the 8th week, when the name changes to from embryo to fetus and all the major organs and systems are in place but in miniature, right? Those cells pretty much are multipotent, which means that there may be some more further differentiation, cellular differentiation, for example, as the brain is forming, etc., the fine-tuning of the various regions that make up the brain. So there is still potential for being any one of those neurological cells in the nervous system, but only within the nervous system. And the same case is made for any one of the other systems of the body, right? Those are multipotent. And then finally, at some point, different cells differentiate at different at various paces, all right? Precisely the neurons are ones, or the nerve cells are some of the cells that take the longest to fully differentiate. But once they fully differentiate, they naturally don't undifferentiate. They don't de-differentiate, they don't go backwards. They stay differentiated until they die, all right? And we can say that the vast majority of the cells in our body are fully differentiated within their particular organ or system and functioning as they're supposed to function. And the proof of that is that we're alive right now and functioning. Okay, so metabolizing, every one of our organs and systems is metabolizing more or less correctly. And so all of the trillions of cells that make up our body are by and large fully differentiated. And so globally, these are the 4 stages, excuse me, the 4 stages of development from totipotent, pluripotent, multipotent, and fully differentiated. Embryonic stem cells. Any question about that? Sorry, question, comment? I probably have gotten too much detail. Do people get it? But okay, so embryonic stem cells are considered pluripotent, okay? They're not totipotent, they're pluripotent because they come from the ICM. But you see that the ICM has the potential to become, because the entire embryo comes from the ICM, then a single blastomere from the ICM has the potential to become any cell of the body, any fully differentiated cell of the body. See, that's where potential comes in. And so embryonic stem cells are pluripotent, and that's why they're considered the gold standard. This will be important, this category, this phraseology of the gold standard is important when we do the bioethical analysis, especially when we contrast them to the adult stem cells, okay? But I can tell you as we're here that adult stem cells are not pluripotent, they are multipotent. See, because first of all, they're being obtained from the born, right? And therefore, potentiality has already progressed, or cellular differentiation has already progressed by the time the baby is born into multipotent. And so it's within particular organs and systems of the body. But they have lost, the cells in our body, by the time we're born, by and large, have lost the capacity for pluripotency. And that's why embryonic stem cells are considered the gold standard, because what's the goal? This happens to me. I forgot to mention the overall goal, really, of the embryonic stem cell research. The goal of embryonic stem cell research is to generate cell lines, see? And there are about 220 cell lines of the body. In other words, the human body, all the dozen organs and systems that form our body, are really supported cellularly by about 220 cell lines. All right, so you can think of, for example, the epidermis as a cell line. I'm just speculating here a little bit. The dermis would be a different cell line. Maybe, I don't know, maybe they have a common cell line, epidermis and dermis, but neurons will be a cell line and so forth. These are like the precursors that make our fully differentiated cells in the body, about 220 cell lines. And it is thought that if we're able to generate about 100 to 150 of those cell lines, right, we could potentially heal any injured or any damaged organ or system of the body. So you can see the potential, the healing potential for that, which is just excellent. I mean, hypothetically, we're talking about, let's say there's cancer of the pancreas. which is fulminating, typically has very little symptoms, and by the time symptoms develop, it's too late. You know, the patient has maybe a week or 2 weeks to live. And so if we could potentially excise the cancerous pancreas and just replace it and grow a new pancreas with the patient's own stem cells, right? And just grow a new pancreas. Then from the cell lines, which would be specialized, would be rather personalized, right? It's a kind of bank. These are Alfred Chalfie's 150 cell line library. Then that has tremendous potential for healing. It's also part of what is known as personalized medicine and all this. Highly experimental today, but that was the goal of these stem cells, of the embryonic stem cells. And precisely because they were pluripotent, the embryonic, then it was thought that one could develop these 100 to 150 cell lines for each individual that needed them going forward in their life. Okay, whereas multipotent is more restrictive because it's only by organ or system, and not all organ and systems. We haven't identified really stem cells, adult stem cells, or born stem cells from every organ and system of the body. All right, and so that's why to this day, the embryonic stem cells, whether human or not, remain this gold standard. All right. Then let me see what I have here. Right. Any questions there before I transition into adult stem cells? I've said already quite a bit about it, but I need to say more. Okay, well, let's go into adult stem cells then. Here we go. So they're known as ASCs. And first of all, they are not pluripotent, they're multipotent, so they're more restrictive in their potential for curing and healing. However, they can be autologous, in other words, obtained from the same patient who needs them. And so there's no immune rejection. That's one of the downsides. There are actually two downsides of the embryonic stem cells. And one is actually more significant than the other. But the one downside, the first downside of the embryonic stem cells is that necessarily they come from another human being, right? Namely the human embryo from which we obtain the ICM. And so think of a patient now who is receiving embryonic stem cells for healing of something. Those embryonic stem cells necessarily come from another human being, and therefore there's going to be a
question. I'm sorry, Father. This is Farid. You said that the adult stem cells is from the same person, right? You said?
Yes, same person, autologous.
Autologous, yes, okay.
Right, autologous, right. So if the embryonic stem cells come from another person, which is the human embryo who was killed, right? Now, those embryonic stem cells are a foreign object to the patient, and the patient will mount an immune reaction against those stem cells. It's just like receiving an organ transplant, right? From another human, it's human, but it's not the same exact genome in the sense that, well, it's the human genome, but it's not the same DNA fingerprint that we each have. And therefore, there's going to be a reaction there. And essentially, the immune system of the patient will detect those human cells from another individual as foreign cells, and will attack them and seek to destroy them, which is obviously something we don't want, right? So the immune system has to be toned down. It can be overcome by toning down the immune system of the patient, right? Just like we do with organ transplants. But it's not the ideal, because now you have a patient who has some kind of illness or disease or injury, And on top of that, we need to tone down their immune system. That compromises them further, right? It compromises them and exposes them to potential infections and so forth. And therefore, it's not the ideal, but it could be done. It could be done in extreme cases if by these embryonic stem cells, the patient could heal, right? Then the toning down of his or her immune system is temporarily until the healing process happens. Mm-hmm. But the bigger issue with the embryonic stem cells is that they actually have too much potential. They have too much potential precisely because they're pluripotent, right? And what we have there is this situation that with pluripotency, because it can become any cell of the body, when experiments have been done in animals, because to this day, there really hasn't been a full human trial that has run its course with human embryonic stem cells, all right? And there's a reason for that. I'll talk about that in a minute. But basically, when these trials were being done mostly in rats, back about, I would say now, going back 5-10 years more or less, it turns out that these embryonic stem cells from rat into rat, all right, they had too much potential. They were actually growing tumors, all right? So here was the scenario. They would take a female rat and take out some mature ova and go essentially through an IVF process. In other words, fertilize her ova in vitro, get to the blastocyst stage, excise the ICM, and grow them to confluence, et cetera, and then once they had a growth of these stem cells expansion, then they would implant. Then they would take that rat, and they were looking specifically at neural cord injury, which was it was the big one, right? If they could heal neural cord, because again, the nervous system is one that essentially does not regenerate, or very, very slowly, practically doesn't regenerate, then that was the most significant healing, right? So they went to that level. Again, they would take a female rat, extract the eggs, fertilize them, obtain the ICM, grow the cells in the lab, and then they would, what they would do to that same female rat now, so that it's an autologous transplant, at least 50%, because the other 50%, once you get an ICM, then you have 50% of the male rat genome there, right? Anyway, there's at least 50% compatibility of the female. They would take that same female and sever the spinal cord partially, sever the spinal cord. They would sever the spinal cord partially to make her paralytic from the hip down. So that, I mean, it does sound cruel, but when you think about, I mean, the protocol was justified for the potential of healing for human injuries. You can think of paraplegics, quadriplegics, okay? So they would essentially make the rat paraplegic, meaning that she would be paralyzed from the waist down, and she could still move. It was important for the rat to be able to stay alive and move, and actually drag herself from the upper limbs, right, and drag her body around. It sounds and looks horrible, all right, but it's not actually that painful. It's not that painful. Okay, this was all done with anesthesia, et cetera, and it's not that the rat is really in pain. You can think of, I don't know if you people know any paraplegic or quadriplegic, and they are paralyzed. I have some friends who are, but they're not really in pain, all right? They're not really in pain from the injury. At any rate, That was the scenario. And then they would take those stem cells of hers and just inject them into the site of injury, into the insult that is called, just inject the stem cells right into the site of injury and hope for the best and see if they would, in fact, rebridge the neurons that were severed on that partial severance of the neural cord around the neck, the neck region. And so as the experiment progressed and went forward, they also had to put the rat on immunosuppressants, of course, a little bit because of the male part of the genome, right? Because the male is foreign to the female body, the male genome. All right, so they did all that with a standard, with the controls and everything else. Everything was, the experiment was very well designed from that perspective, and let it go forward. And within days, what happened was that the embryonic stem cells did start growing, but they started growing as a mass, all right? They did not differentiate fully into the neuronal cells, into the fully differentiated neuronal cells that were needed for bridging the gap, bridging the site of injury. Instead, they grew as a mass of cells, which was equivalent to a cancerous mass, And in retrospect, it makes sense biologically, because when you think about it, the whole embryo comes out of the ICM. And so it is like the whole embryo was trying to grow there on the back of her neck. You see, the whole embryo. In other words, they had too much potential. Bottom line, they had too much potential, and the whole embryo was trying to grow on the back of her neck there because they had too much potential. Okay, so the silver lining about adult stem cells being multipotent is that they are already system specific or organ specific, and therefore they actually perform better in trials, in actual clinical, human clinical trials that have been done. And so the adult stem cells are actually performing better than the embryonic stem cells, okay? And that's why, to my knowledge, the only one that was able to, the only biotech that was able to secure a protocol for embryonic stem cell research from the FDA was Geron. Geron is a biotech from California. That was one of the first biotechs in the US, goes back decades, and is kind of the pioneer in this area and many other areas of biotechnology, okay? And Geron had secured a protocol for, I think it was macular degeneration, which you can see is not even a life-threatening condition, all right? They just wanted to have proof of principle. Proof of principle was that human embryonic stem cells would work in some healing of some kind of human injury or defect or illness, all right, or accident. And so they chose macular degeneration, which is not insignificant. That could eventually, it leads to blindness, right? But blindness is still compatible with life. Certainly blind people can still have a full life, no question about it. So they chose that. And they tried, because there is the Institute for Stem Cell Research in California, which has actually state funds in the tune of millions of dollars, right? So that institute has been going on, and they were trying to, for years, I mean, this goes back into the 90s, they had a proposition that was put out for the citizens, and the citizens basically voted in favor and so forth. And to this day, they didn't have really any tangible results of success after millions and millions of dollars of research into this area. So they were kind of desperate for getting some result, and they partnered with Geron, et cetera. So they started the trials, and within, I don't know if it was weeks or months, but some problems started arising precisely in one of the clinical trials. Some problem arose with respect to the embryonic stem cells that were implanted into this one patient who was a candidate for the trial, okay? And so they suspended it. They suspended it and they pulled their permit from the FDA, and that was it. Now, of course, some of this information is confidential, classified, because it's you know, it's not copyright, but it's anyway, it's privileged information of the company because it can be patentable, right? And therefore they have the right to keep some of the information private, but in the bigger picture of the overall protocol, they have to be transparent because they're using public funds. And even if they weren't using public funds, they're being regulated by a public agency, which is the FDA. And so they have to have some level of transparency, especially if the experiment is not going well, all right? So they did, they had to rescind it. And apparently it was this whole thing that just the embryonic stem cells have too much potential, and they tend to grow tumors instead of actual healing, all right? So you see, that's the silver lining about adult stem cells, is that because they are precisely toned down, they're multipotent, then they're showing more success. And here's a website, actually, stem cell research, that was generated back then. This has been going on for several decades now, and has been monitored by a group of scientists and actually philosophers and theologians. Stem cell research. I think it was .org or .com, maybe .net. So research, let's do that. Anyway, I don't know if it's been taken down or this site was available.
Father, I couldn't access it either.
Yeah. Okay. Well, this is my bad. I should have checked this beforehand. I just assumed that it was still on. Anyway, this is it's a site that was monitoring the all the success that was going on with adult stem cells. Let me go back here. Maybe they changed their name, their site, I don't know. Oh, okay, well, they had another name. It's a group of scientists. It's called Do No Harm. They've kept a site. Bear with me for a moment here. Oh, that's a film. My bad. I should have checked this site beforehand. Anyway, so this site, basically what they had was a list of trials at different levels of success, from peer review articles, scientific and clinical articles and so forth. And at that point, they had about 70 different success stories at different stages of development with adult stem cells. And to that date, no success in human clinical trials with embryonic stem cells. So it was very informative. I apologize. I need to look at this further, but it was a very useful site. But I mean, it doesn't depend on them. We can just punch here adult stem cell research. Okay, so here's from Mayo Clinic. Just looking at it from the first set, only recently researchers thought adolescents could create only similar types. Emerging evidence suggests that adult stem cells may be able to create various types of cells, right? Bone marrow, heart muscle. Okay, so you can see this current site in a Mayo Clinic, which is internationally renowned, and they are essentially saying that there's potential here. Okay, I don't want to take time to read it in detail.
This is Farid again. Sorry, well, you said that it made success at various trial levels. Do you know more or less the percentage of success of this use of adult stem cells?
No, I don't. But I mean, again, now this gets into anecdote, right? Just last week, for example, I had a parishioner call me and ask me a question because they have arthritis of the knees, and this person was going into the hospital to extract some of her own stem cells for being injected into the knee. For there is an experimental trial apparently for curing arthritis of the knees or of the joints with autologous adult stem cells. Mm-hmm. And it's experimental, you know, and she wanted to know if it was ethically okay and so forth. And she explained to me a little bit of the protocol. It's basically pretty much they needed her consent to do that. And she wanted to make sure because these are stem cells, Father. And I know she said the church is against stem cell research. Okay. And so I clarified, I didn't go into a 2-hour lecture, but basically they were her own stem cells. And as long as she consented and had some kind of promise, right, it was okay. So I know it's going on right now, but I really don't know in detail percentage of success and so forth. But it is going on for sure. I mean, we just saw it in the Mayo Clinic site, right?
Yeah. Thank you.
Okay, so it's there. It's there. And it has to do precisely, I mean, the core of it is this, that they are multipotent in contrast to pluripotent. So in this case, less is more. All right, this is one of these classical cases where less is more. Less potential is more benefit, whereas more potential is less benefit because they're just wild tumors, which, you know, the definition of a tumor is basically uncontrolled self-growth, right? They don't respond to, they don't respond to confluence, they don't respond to signaling, cell signaling, etc. All right, forward. IPS. Now, this is Shinji Yamanaka. I don't know if you remember Yamanaka, but Yamanaka is a Japanese researcher. And this was relevant to me when I was at Purdue precisely because it was happening in the early 2000s. I mean, it's been 20 years now, it's been 2 decades, but back then it was fresh. And I thought it was exciting that as I was doing my research at Purdue, this was going on, that he was in the news back then. And anyway, what happened here was this researcher was in one of the universities there in Japan. I forget if it was, what do you know, Osaka. Okay, it was in Osaka. At any rate, he was doing embryonic stem cell research, human embryonic stem cell research. Okay, the race was on by throughout different labs of the world back then of who was going to be the first one to be able to tame these human embryonic stem cells and to eventually develop these cell lines, all right, the human cell lines. And it was a little bit, it was kind of a deja vu of the Human Genome Project throughout the world, who could get that human genome first. And so it's kind of a competition going on throughout labs of the world. And I remember the interviews, okay, because Yamanaka is the one who invented this technique of iPS stem cells, induced pluripotent stem cells. Keep in mind, see, pluripotent, right? In other words, he wanted to have an alternative to the, human embryonic stem cells that did not involve killing human embryos. Okay, now think of this. He wanted an alternative of human embryonic stem cells that did not involve killing human embryos. And I remember this distinctly because it was in the news at the time, and when he finally came up with this IPS, he was interviewed, and of course it was all over the world in the news. You can go back 20 years and look at the news back then. And basically he said that as he was doing his human embryonic stem cell research, and he was disaggregating these human embryos. Yeah, so where were the human embryos coming from to begin with? Obviously, in vitro clinics. These are the spare embryos, right? These are the spare embryos in in vitro clinics that were being donated to science. Remember how I talked about that? That the donation was a technicality. First of all, the clinic was being reimbursed for the expenses of generating those human embryos and preserving them until that stage of donation. But more importantly, when he was looking at these human embryos under the microscope to disaggregate them, right? He thought to himself, and he said this in an interview, he thought to himself, these human embryos are looking very similar to the way my daughters looked at that stage. I think he had three daughters. And this is what he thought when he was disaggregating these human embryos, these blastocysts. He said, these blastocysts look very similar to the way my daughters looked when they were that stage. So, that is amazing. That is amazingly honest from this researcher. Here we have a scientist who is top of the line in his research, and he's generating, he's trying to generate these cell lines from human stem cells. And he's actively thinking. He says, these are essentially human beings, just like my daughters were at this stage, and like I was, and any one of us at this stage. And so he said to himself, there has to be an alternative. There has to be an alternative to disaggregating the human embryo. In other words, leave the human embryo alone and let's see how we can undifferentiate, de-differentiate, right? Differentiated cells, and then redifferentiate them again. So of course, this goes against nature. This part, the undifferentiate or the de-differentiate, right, is going against nature in the sense that he's going to take differentiated cells and undifferentiate them back to the pluripotent stage. In other words, he wanted to go up the hill here, taking fully differentiated cells, right, and moving them up, but stop here, not go to pluripotency. Stop here at pluripotency. And of course, not to generate a blastocyst. He wasn't about generating a blastocyst. No, he was just generating individual blastomeres. He wanted to generate blastomeres that would be equivalent to an ICM, but not the whole ICM. And so you see the ethical distinction here. It is splitting hairs, but it is splitting hairs that are not insignificant. He wanted to undifferentiate differentiated cells to the pluripotent stage, but individually, individual cells, not the ICM as a whole. So by doing that, we don't really have a human being. There's not an ICM mass as a whole. We just have individual cells of the ICM. So you see, it would be a way of obtaining individual ICM cells, totally artificially, of course, but possibly ethically, because we're not destroying a blastocyst. We're not destroying a blastocyst at all. So we're not killing a human being. All right. In other words, his idea, as impossible as it sounds, could be justified ethically. The whole issue was here in the to undifferentiate, fully differentiated cells. And then, of course, the second stage was to redifferentiate them into a different tissue or the cell lines that I've been talking about. All right, brilliant, brilliant. Of course, you know, these ideas that are not intuitive, because as far as we have seen and observed in nature, as far back as when the cell theory first arose, right? That the cell is the basic unit of life, and scientists have become aware of this whole issue of differentiated cells, which are functional in any body, not just a human body, but any living body that we see, animals, plants, etc. And so this idea of going back to undifferentiation, it was just not thought possible. It turns out that there are regulatory genes, there are regulatory factors. The factors, right? The regulatory factors, which are essentially proteins that regulate this process, right? The regulatory proteins that regulate this process, of course, they are the product of regulatory genes. So some genes kick in before others, correct? And regulatory genes have that property. They establish the basic body plan and so forth. And they do that through factors. In other words, through proteins that are the ones that are doing the actual work, the chemical signaling necessary. And so him and his lab started working on that to find which were these regulatory factors. And it turns out it came down to about 4 factors, really 4 proteins that are involved in that cascade of differentiating cells. And so, and there was a big race, again, to find what those regulatory factors were. And he was the first, and he himself said, you know, he was working day and night. He had his lab working amazing. He worked out a schedule where essentially the lab was going on all the time. They would sleep very little and take turns and so forth. And with his research team of postdocs and grad students and whatever his team was, they worked on this. Essentially, they were looking for the regulatory factors that were involved in cellular differentiation. And it came down, I think, to about 4, if memory serves me, 4 basic proteins that were necessary to be present for the thing. So if any one of these proteins were missing, right, then, or added in a different, with a differentiated cell, that cell would start undifferentiation, will start becoming less differentiated, okay, will revert. And specifically, he used human fibroblasts, which are part of the mesoderm, the germ layer, okay? And he got them to undifferentiate and then redifferentiate into the 3 different types of muscles, which are skeletal, smooth muscle, and cardiac muscle, right? The 3 different types of muscle tissue. He did that with fibroblasts. And so he was successful, and he was the inventor of this, what is now known as IPS. Some technical issues where essentially the needles that he was using to inject these factors into fibroblasts were viral capsules. Of course, imagine, so we're dealing here at the microscopic level right? And how do you inject these factors, in other words, these proteins into cells, maintaining the integrity of the protein, in other words, the conformation, the four-dimensional conformation, so that the protein is active, right? Inject them into individual cells. Well, he used viral capsules, which is not the first time. That's kind of a standard procedure in the lab for injecting factors into cells. Use a viral capsule essentially as a vehicle, as a hypodermic needle to inject these proteins, all right? And the challenge with that is, of course, these capsules had to be inactivated, right? Because you don't inject the cells with the fibroblasts with actual viral sequences. You want to inject them with a protein that you're going to put into that capsule, which the capsule typically is a protein shell, right? So the technical challenge was, and this is done in shotgun type of protocols. So if some of the viral capsules still had some functional DNA or RNA, I don't know if you used DNA or RNA viruses, that could contaminate. If there was some viral capsules that still had at least some of the viral DNA in it, that could contaminate, the fibroblast and give a skewed result and actually infect the cells with the virus and so forth. So there were issues there. Or if that viral DNA could somehow affect and influence the factors, the human factors that were being used, the regulatory factors that were being used for undifferentiating the fibroblasts, all right? So there are a number of technical things there. I understand that those issues have been worked out now, and these factors can be used safely to undifferentiate human cells and redifferentiate them again using the IPS technique, all right? But the I stands for induced, and induced is the reference to the artificiality of the process of the going backwards, the undifferentiating of the cells. But essentially, he was successful. He was able to generate pluripotent cells that were not obtained by destroying human blastocysts and had essentially the same quality as ICM cells. Mm-hmm. Now, I haven't really kept up with the research to date, but it's out there. I believe it's still at the experimental level, especially because since then, there has been an increasing number of sources discovered for adult stem cells in the human body, the pulp of baby teeth, the hair follicles, bone marrow, adipose tissue. That's why I say the liposuction is full of stem cells, adult stem cells of that customer, right? So if I cut myself, that cut is going to heal from the inside out, right? And where are the new cells coming from? Well, they're coming from stem cells in my dermis or adipose tissue that are there waiting to heal in the case of an injury. And even the blood which is the most sweet discovery, if you will, that human blood has in its serum, has some stem cells floating around. They're hematopoietic, meaning that they come from the blood system, right? The mesoderm. So in that sense, they're similar to these fibroblasts. They come from mesoderm, essentially from the bone marrow. But they are stem cells, and they're just circulating in our system. They're typically few, all right? So they're not that many in quantity, and therefore they need to be amplified in the lab and so forth, but they're there. And to obtain them, basically, it takes like something equivalent to a blood donation or something like that, or maybe will be processed more akin to pheresis. Too much detail I don't want to go into, but to obtain platelets, for example, that are also free floating in the serum. Pheresis is a technique that is used for obtaining platelets. Similar to that, I presume, is obtaining these stem cells, our own autologous stem cells from our blood. All right. And so that bypasses, again, just bypasses the immune system, immune rejection, and there are the successes that have been going on, etc. Any questions about the adult stem cells? All right, there's another category that is kind of a hybrid, for lack of a better word. Technically or physiologically, they're embryonic in that they come from the newborn, but you know, anthropologically or even legally, they come from the born. They're adult technically because they're from the newborn. And this is what I'm talking about. I'm talking about cord blood cell, all right? Cord blood stem cells. So there's the umbilical cord that is in between the placenta and the newborn, right? And once the newborn is born, right, what happens to the umbilical cord generally? It's medical waste. Generally, you know, it's cut, severed, and the part that is attached to the placenta is discarded as medical waste. And the part that is on the baby side is knotted and pushed into the belly and put a little tape on top of it, and that becomes the navel, right? The belly button. So the umbilical cord basically is discarded, but it has blood in it, and it has blood that belongs to the baby by and large. Okay, it's on the baby side, it's on this side, it's on the baby side of the placenta. And so that cord blood, just like our own blood, has some stem cells in it. Has stem cells in it. And those stem cells are pristine. And from one perspective, from several perspectives, they are ideal stem cells because, yes, technically they're adult stem cells because they come from the born. So even stem cells from a child, from a chronological child, are considered legally adult stem cells simply because the child is already born, right? It's not an embryo anymore. The same with these umbilical cord stem cells. Technically, they are adult stem cells. However, because they come from the umbilical cord, they really are more embryonic-like, in the sense that they are pristine. They haven't really been exposed to life outside the womb for long. You know, just consider UV exposure, for example, over the years, etc., and even just age, the senescence of cells, how cells in our body age and so forth. And so they're pristine, right? They have the potential for regenerating basically any organ and system of the body. So technically so what do you think? Are they multipotent or pluripotent? Remember, pluripotency stops with gastrulation, okay? So definitely they are multipotent. In fact, they come from the bloodstream of the embryo. So they're part of the circulatory system. But they're very early on in development, and therefore they're very tender. They have plasticity, right? They're able to heal well damage. So potentially they have the capacity for healing all these different organs that are illustrated here. And they're known as MSCs, mesenchymal stem cells, mesenchymal, because they come from the mesoderm or the blood system of the baby. Okay, and this is when you hear couples saving the cord blood of their newborn, and it has to be done precisely there when the baby is born. They have to do the whole process there because that's when the blood is pristine and before it starts spoiling. The way to do that is they can even just go online and buy the kit that is going to be used, basically the syringe and the bags and so forth that are in sterile environment, and they will give that kit to the nurse or the attending, whoever is going to be involved in the there at the birthing. Of course, they want to tell their OBGYN also so that everybody's on board. All right, but the basic idea is that when and the whole, let's be clear here, the whole emphasis of the labor is focused on giving birth, all right, not obtaining the stem cells. So this is secondary. If and when everything goes well and the placenta is, the umbilical cord is there available and so forth, they'll do the procedure, but the focus obviously is all on giving birth in a healthy and safe way. All right, so you can imagine the scenario, the mom gives birth, and now we have the umbilical cord and the placenta there. The nurse will simply insert the syringe and extract the cord blood, put it into the vials that are provided, and send it to the lab, right? What is done at the lab is they don't actually save the whole cord blood. They do the isolation right then and there. They spin it down and isolate the stem cells, and that's what is preserved. And how is it preserved? Well, cryopreservation. So at the end of the day...
Sorry, Farid, again, I'm sorry to bother. You repeat the process. They take a syringe and they extract the blood?
Right, they start the cord blood after birth, all right? They extract the cord blood and then send that sample of cord blood to a lab that is already pre-labeled and all that. And then in the lab, what happens is they spin down those that blood that's separated, right, and extract the stem cells from that fraction, and then put them in cryopreservation. The stem cells are then cryopreserved for future, and the hope is that they will never be used, right? that the baby lives a full lifetime and lives to old age and beyond and so forth, and there was never any need to use them. But if this baby going forward at some point her or his life sustains an injury or an accident, whatever, or an illness, and he or she qualifies for an autologous transplant, then the cells are available there, cryopreserved, you see? Remember, they're in suspended animation, right? So in principle, the time factor shouldn't affect them if the metabolism, what happens with cryopreservation, essentially metabolism is slow to a halt and everything is just suspended, but they're alive. They're alive in suspended animation and so forth. Not only can the donor use them later in life if needed, also some siblings, because a sibling would be compatible to some percentage, right? Again, it would be equivalent to a transplant, a sibling transplant donation, but of stem cells instead of a liver or a lobe of the liver or a kidney or something like that. It's equivalent to an organ transplant, and so siblings could benefit, and perhaps parents could also benefit in their I don't know, old age or something from those stem cells. So you see that those MSCs have potential for a number of people in the family, not just the donor herself or himself. Okay. So why isn't everybody doing this? Why isn't every couple preserving this, their cord blood of their babies? Well, for a number of reasons. The first reason, sadly, is always ignorance. They just don't know that it exists, but it's getting around more and more. And then the second, a close second is finances. It's expensive. Well, it's some expense. I understand that the initial procedure, obtaining the kit and doing the procedure and so forth may be $1,000 or $2,000 or $3,000 between buying the kit. I think the kit itself is not that expensive, but whatever the hospital is charging with overhead of doing this runs into several thousands, maybe 1,000, 2,000, 3,000. I don't know. Maybe someone has done it recently and can update me on the price. But then, so that's one part. And the second part of the expense is just the maintenance. In other words, keep in mind, these cells are being maintained by some kind of a lab. It could be precisely an in vitro clinic that is holding them, or BEMIDAT, it could be somewhere else, some other storage, but some kind of cell storage that does cryopreservation in a professional way, right? With all the guarantees that can be offered with modern technology. And so they have a price. They're not doing it for charity. So that's these biotechs are for profit. So they charge, typically what I've heard is like maybe $100, $200, or $300 a year of maintenance, right, for cryopreservation, which is okay, but it depends on the finances of the couple, especially if it's a young couple with a firstborn and so forth, and they're still struggling with the finances. And it adds up over the years. It's $100, $200, or $300 that has to be budgeted just for maintaining those cells out there. So it adds up. And from what I understand to this day, insurance does not cover it, unfortunately, because it's considered an elective procedure. So my argument on all this is that couples should group, band together and request from their insurance companies to cover it, or at least cover some percentage of it, because in my mind, it behooves insurance companies to cover this procedure, because it may save them money in the long term, because this may be a less expensive way to heal an injury or an illness down the line of the insured. Let's say the baby is insured and then they sustain some injury years into their life. And let's say it's a serious injury that will cost X amount to the insurance, typically in the thousands, maybe hundreds of thousands of dollars. I don't know, depending on the severity, right? But if they have stem cells stored, maybe those stem cells will reduce the cost of healing, right? Because of their potential and so forth. So I'm thinking that the insurance companies could benefit eventually from more and more couples doing this. Not only that, I understand that now there's a national registry where these cells are genotyped and put on a bank as if they were organs, right? Because they are somehow analogous to organ transplant. And there's a registry, a national registry, where people could do matchup on compatibility. And we don't have to reinvent the wheel. There is already a system elaborated that is very elaborate, actually, in organ transplantation for matching up a donor with recipient and so forth on genotyping. So anyway, I think it has potential. I don't really know why the insurance company is kind of reticent to cover it, but they probably go through some protocol, and unless they see more results, more benefits in the area of adult stem cell research, then they're still, I guess they've done their cost benefit analysis. It still doesn't add up for them. Maybe, I don't know. It's an option. I just mentioned it because it is an option that is viable for couples who want to go this route. And as far as the ethics is concerned, of course, what's the big issue here with the ethics of MSCs? What do you think is the one issue really, ethically speaking? It's so obvious that people miss it.
The ethic with what, you said?
What is the ethical issue with MSCs? It's so obvious that people miss it. Consent, just consent, you know, that the couple needs to agree to this. Obviously, the newborn cannot agree or disagree, right? But it is definitely the intention. So what's the intention? The intention is potential healing for the baby or someone else to benefit, right? So the intention is definitely a good intention, laudable. Now, how about proportionality? Is the life or the health of the newborn being threatened by any way, in any way? No, not at all, right? The baby is already born. They're about to discard, they're about to incinerate this umbilical cord with its contents. So there's no damage to the baby for sure, or to the mother for that matter, or the father. And yet, I'll tell you, I mean, you know, ignorance is we have to spend our lifetime pushing back on ignorance, okay? Because it is horrible. Going back to the little anecdote that I was about to say at the beginning, I'll say it now. At St. Elizabeth Medical Center, where I was doing research for about an hour, about a year, back 20 years ago, one time I was talking to one of the nurses in the cafeteria, just chatting, one of the nurses there, and she was she was a delivery nurse, okay? And I asked her, does the hospital do umbilical cord stem cells? Do they preserve, you know, do they extract, do they rescue these umbilical cord stem cells? And she said to me, she happened to be a Filipino nurse, you know, which many of them are Catholic and they make very good nurses and so forth. Anyway, and she knew that I was a priest, and so we were chatting, and she says, "Oh no, Father, we don't do that here because this is a Catholic hospital." This is a nurse who is a delivery nurse, okay? And I'm thinking, oh my goodness gracious. Think about this. You know, obviously when, I don't know if this topic was obviously not covered when she studied nursing way back there in the Philippines. They come with their nursing degree already, and apparently it's you know, it's one of the degrees that is just recognized in the US, and they go right into the workforce. So I said to myself, look at this. She really doesn't understand or has never considered that this is ethically okay because the baby's already born and we're just going after the cord blood that is about to be incinerated. So, yep, this is the world we're living in, and that's why ignorance is like darkness. Unless we shed a light, there's darkness. Unless we turn on the light, it's dark at night. And unless we inform people, we continue ignorant from birth forward. Unless someone takes the time and the effort and the love to teach us something, we remain ignorant. And therefore, once we know something, we have the responsibility to teach that something. All right. All right, it's been 2 hours. I need a throat break. Any questions or comments before we take a little break here? Great. All right. I can't believe that you've understood everything I've been saying, but be that as it may, I'm going to pause recording. It's 11:26. Okay, let's be back by Human cloning, let me see. Okay, 10 minutes, okay? 10 minutes, 11:30. I'll start at 11:36. Thank you.
Thank you.
Okay, my dear students, welcome back. We're going forward now with human cloning, hopefully. Any questions or comments on the previous stuff? Just go back to the original slide. Right, so we covered the distinctions between human and non-human, adult and embryonic, cord blood, iPSC. Now we have the topic of human cloning to cover. So if there are no questions or comments, I'm moving forward here. All right, this is also at this point a little historical, and sometimes even hysterical, as you'll see in a minute, because we have to go back about 30 years, more or less, when we talk about human cloning. It was in the 90s, I believe. And any of you remember Dolly, the sheep? Not the singer, but the sheep, even though there is a connection.
Yes.
Yes, some of you may remember. Here's Ian Wilmot, who is a researcher who cloned Dolly. This is Dolly. This is actually taxiderm Dolly. All right. She was eventually euthanized, and you'll see why. But this is actually the euthanized animal, which was taxidermed and preserved for posterity because of her claim to fame of the first cloned mammal. So the first cloned mammal, get to it in a minute, was Dolly in the 90s. And I can tell you also, I mean, I'm inserting these personal anecdotes in here because I don't know, I find that maybe it will help other people, motivate other people to further studies, right? Because it was in the 90s and I already had my doctorate in moral theology from Rome. But at that point, my bachelor's in biology was about 25, getting close to, you see, yeah, about 25 years old. So already I was doing bioethics. I was a bioethics consultant in the Archdiocese of Miami and other places, even at the national level, with an outfit called the National Catholic Bioethics Center. One of the things that we do in the NCBC is to do consultation for the bishops of the United States in bioethical issues and so forth. So I was already involved in bioethics in the 90s, but I was realizing that biology kept advancing and biotechnology, mostly biotechnology, advancing to where things that used to be science fiction in the 70s when I was studying biology at FIU, my bachelor's of biology at FIU here in Miami, now they were becoming a reality in the lab. Okay, and one of them was this cloning that just struck me when I saw it in the news, the cloning of the first mammal, who was this sheep, Dolly. And I said to myself, well, you know, mammalian cloning in the lab, the technology for mammalian cloning is essentially the same for a sheep or a dog or a cat or a human. It is what is known as somatic cell nuclear transfer or just nuclear transfer, right? And therefore, is anytime now before any lab wants to clone a human. And there's certainly a bioethical difference between cloning a human or cloning a non-human, right? The bioethics, even though the technology is the same, essentially, right? And so I said to myself, I better update my biology because at this point it's just ancient. And that's when I went to the Archbishop of the time, Archbishop Favalora, and asked him if I could go ahead and update my biology degree. And that's when he said, sure, go right ahead. What do you want to do? I said, well, I'd like to do a doctorate in genetics because that's the field to this day. That's the field, right, where they can have more controversies simply because of the technique. Think of CRISPR, for example, where we can now insert essentially any gene, any genetic sequence from one species into a targeted area, not shotgun like it used to be done, but into a targeted area of the genome of any other species, right? It is amazing the power of CRISPR today in the lab, which was just pure science fiction a few years ago. And so that is not insignificant. That is not without its ethical issues. What genes that we're introducing from what species into what species, you know, and for what purpose. So definitely needed to update, and that's why I thought that genetics was the best way to do it. And that's when I went to Purdue, and I can just tell you, I have no problem in telling you that I was 50 years old back then. And so I obtained a second doctorate in genetics, totally scientific, right? Empirical, to the point that, as I mentioned, I had to prepare for GREs and all that, and just get accepted into graduate school like any other student. And it can be done. It can be done. And so I'm telling you, if anyone tells you, or you start thinking, oh, I'm too old to study anything further, not acceptable. Not acceptable. In fact, as you know, we have a number of seniors in our program that that you're sharing classes with, all right? And so these seniors, I salute them because they, some of them are close to contemporaries to me, and yet they're biting the bullet and getting updated. So the best teacher is always a student, and the best student is always a teacher. And we need to update, and sometimes we need to update formally by an actual degree that gives us that competence. Therefore, there we are. Anyway, this is one of the things, Dolly the sheep. The news of Dolly the sheep is one of the things, key things that motivated me into asking for a second bracket, if you will, parentheses of my priestly life into obtaining a terminal degree, you know, and I frankly feel extremely privileged that the Archbishop said yes for me to do this. And so I feel the responsibility now to give back. And that's why also I generated this master's in bioethics, the Master of Science in Bioethics at St. Thomas University about 6 years ago, because it's one of the paybacks, if you will, right? It's putting the knowledge to work. There's no other university, certainly in Miami, that has a Master of Science in Bioethics like this one, especially from the principled perspective, or in the state of Florida that I know of. I've gone through the curricula of the various universities in this area and so forth, and I haven't seen anything in, at least in the Southeast region, that is comparable. Okay, so there it is. Back to cloning. Functionally, for the purpose of human cloning, because we have to ask the question, you know, why? Why do this, right? It's not that elaborate of a technique, but it's certainly very impactful to generate a clone, a human clone. And there are even science fiction things about this. It's a number of movies, science fiction movies. Some of them actually get pretty close to reality. But essentially the why, the motivation, the intention is one or the other, either for reproductive purposes or for therapeutic purposes, reproductive or therapeutic purposes, right? Okay, so basically reproductive purposes is for a couple who cannot have children to have a child through cloning. All right, so it's kind of an alternative to IVF, even though it uses a lot of the IVF technology. But it's an alternative, and you'll see how. But basically, it's for a couple in the, let's say, in the simplest case scenario, to have a child, and therefore that's reproductive. Whereas therapeutic is not for the purpose of generating a child for a couple to have a child, but actually to generate human embryos precisely to extract their stem cells, all right? So in other words, a source of human embryos that does not involve fertilization, if you will, all right? And therefore, by in the minds of some people, that zygote that is generated, because that's what happens with human cloning or with any cloning, essentially we generate a zygote, okay? But we generate a zygote with a somatic nucleus, not by singamy. And so in the minds of some people, that's not a human embryo somehow, but that's not creating a new human being, okay? And so because that blastocyst is not really going to be implanted or anything like that, it's just being generated for the purpose of obtaining embryonic stem cells. So really, they were just after generating ICMs, if you will, viable ICMs. In other words, human blastocysts. Right? So those are the two main reasons for human cloning. And in fact, here is a little, another micrograph again. Here's that vacuum that I talked about earlier, okay? Let's just amplify this one a little bit for detail. Here's the micro syringe, the micro pipette, that little glass syringe. Here is, in this case, this is a human ovum, or an ovum, just in general. Let's say it's a mammalian ovum, okay? This is a mammalian ovum, excuse me, that has been enucleated, And so basically it's just a mammalian cytoplasm of an ovum, because it has to have the factors in the cytoplasm for development, precisely the regulatory factors that I talked about, including those 4 regulatory factors of early embryonic development that are in the cytoplasm itself, okay? So this ovum has been enucleated, and now it's being nucleated with a somatic nucleus, and you can see it right here. Here's a somatic nucleus that is being injected into the enucleated ovum, all right? So that's why it's also known, well, the full term, so here, full term is somatic cell nuclear transfer, SCNT, all right? So it's a nuclear transfer. In other words, the egg nucleus was taken out, enucleated, sucked out, right? And a somatic nucleus is being inserted. Somatic simply because we need a diploid cell, right? The zygote is diploid, so we need to generate essentially a diploid cell. And so it's also known commonly now is sometimes you just see NT, nuclear transfer, all right? And it's assumed that it's a somatic cell nucleus that is being transferred. So sometimes you just see NT for cloning. All right, so the nucleus that is transferred in, right, is diploid. And therefore, the zygote that is generated, or the cell that is generated, is a diploid cell. And it's a single cell, and therefore it's equivalent to a zygote. It's functionally equivalent to a zygote, right? You can see the somatic nucleus as the equivalent, the functional equivalent of the two pronuclei fusing during fertilization up to syngamy. Now we have a diploid nucleus in a cytoplasm that comes from an egg. So again, the factors, regulatory factors are there. Okay, so it turns that ovum into a new zygote functionally. And then that new zygote just is allowed to move forward and differentiate and go through embryonic development. In other words, if everything goes well, that zygote will turn into a morula, and the morula to a blastocyst. Then the blastocyst is implanted into a womb, and then allowed to continue embryonic development, and hopefully, in the case of a human, of course, 9 months later, the baby would be born, right? That would be for reproductive purposes. But if it's for therapeutic purposes.
Shofi, I have a question. This is Farid.
Yeah.
In the case of Dolly, I remember that the implantation was done in a foster sheep. So yeah. I don't know why they didn't do it in the same.
Right.
If that donate the ovum.
Yeah, it's basically proof of principle. Just one slide away from your answer, okay? It's coming up on the next one. I was just trying to explain in too much detail what cloning involves. You can just read it in Wikipedia. It's a very simple explanation. Let's go to Dolly. Okay, so this was done at the Roslin in Scotland, again in '96, and this is how he did it. He had an ewe, I think this is called a ewe, right? A female sheep. Donate the egg, right? Yes, through, I don't know if they did hyperovulation or not, but they obtain, excuse me, sheep ova. And, oh, sorry, here's the egg. And they extracted the nucleus, they nucleated the ova, all right? Let's stick with one ovum, nucleated the ovum. So here's the enucleated ovum. And then in this case, they actually fused, they didn't even bother doing the intracytoplasmic nuclear injection. They did an even more crude technique, which was by zapping the two cells together, right? And this can either be done by electrical charge. Typically, it's an electrical charge. Yes, they obtain a donor nucleus, but actually they took just a whole cell, just a somatic cell. This is a somatic cell from another sheep, all right? And they used the udder of the sheep. There's a reason for that. The udder is the mammary glands of the sheep. Seemingly, these mammary glands are pristine. They're the tissue there, the epidermal tissue is easy to obtain. They're obviously somatic cells, right? Because that's a differentiated organ. And they wanted it to be a female. So this is an easy way of obtaining these cells from a female without significant harm or injury to the ewe. So they fuse these two cells together, the enucleated ovum with the somatic cell, to get that nucleus, the somatic nucleus, into the ovum, right? So here's the fused cell. And essentially, so this is functionally the same as a zygote, in that it has somatic nucleus and then the developmental factors in the cytoplasm of the ovum. And this is in the lab. So this all from here forward is in the lab, right? At 37 degree, incubate. I don't know how long it takes for the sheep zygote to develop into a blastocyst. I assume it's around the same amount of time, more or less, maybe faster. So here's a little bit of a misrepresentation because this should be a blastocyst and not a morula. It looks more like a morula to me, right? But it's the blastocyst that they want to implant back into the mother. And they use, in this case, they use the same ewe from which they took the ovum. So these two sheep are the same. All right. Here they're looking at compatibility. They're just trying to give that embryo the most hospitable place, the most hospitable uterus possible, knowing that already the nucleus is coming from another ewe. Why they used another ewe will become clear in a moment. Let me just follow this one through, at least until implantation. So you understand that they take that blastocyst and implant it into the sheep that donated the egg. All right, so there's compatibility there, at least at the cytoplasmic level. And then they just weigh out write out the gestational period, which I don't know how long it takes for a sheep to develop the embryo, but whatever time it takes. And she will give birth to Dolly. Okay. Now, let me go back here. Why this one? Because they didn't want to keep everything within the same sheep, because that would be not proof of principle. It would be kind of proof of half principle. In other words, if the somatic nucleus still comes from the same sheep, then that's an autologous clone. You know, that's a self clone. That's a clone of self, but it's not a clone of other. But the proof of principle, the full proof of principle, is that the clone is a clone of other. In other words, Dolly is identical. So by definition, clones are identical, right? They're identical twins. They're monozygotic twins. So Dolly is a clone of the gestational mother, or is a clone of the somatic nucleus donor? Which one?
The somatic, no? The somatic.
Yeah, of course. In other words, who's directing the genome here? Who's directing the genome, the development? It's the genotype, right? It's the genome in the nucleus. So the fact that the mother's, well, who's the mother? Okay, the gestational mother, the surrogate mother, the gestational mother, the fact that the cytoplasm of the egg came from her, from the gestational mother, that just makes her the gestational mother, okay? Regulatory proteins, yes, but the nucleus comes from this donor, and that's what's directing the actual development, all right, into this individual lamb. So this lamb, Dolly, is an identical twin of the donor somatic cell donor, all right? Even though they're years apart of age, but it's an identical to this guy, to this gal. And that's the full proof of principle. In other words, what they wanted to prove with this is that cloning will work essentially with any somatic cell of the same species. Because if you just keep it in-house, if you keep it within the same animal, same individual, you've only proven the principle halfway, but not all the way. So that's why.
Okay, thank you.
You see why it makes sense, okay? Now, I mentioned, so this is reproductive, okay. So, and the idea behind this, really what was motivating Wilmot and others at that time, and again, that was raised with different labs of the world, but he's the first one to do it and publish it in peer review. So the idea behind it was precisely to generate these embryos that could be used for obtaining embryonic stem cells. Okay. Again, the idea was that if this was used for somatic, for therapeutic cloning, in other words, to generate human embryos, translate the model into human cloning, embryos could be generated, but they would not reimplant. All right, this just proved that that embryo was normal. In other words, the fact that Dolly was born and lived as a sheep until she was euthanized prematurely, and I'll get to that again, but the fact that she was at least born and looked normal and ate grass and lived normally for a few years, proved that these embryos were normal. Okay, that the embryos generated through cloning were normal. So that was the whole thing. But now the idea was to use human cloning for therapeutic purposes. In other words, to generate zygotes or to regenerate blastocysts, more specifically ICMs that were human and that would have imagine the potential for healing. Hypothetically, one could use the cell, the somatic cell could come from the patient who is ill. So you would have autologous. You, in principle, that those ICMs, that ICM would be fully compatible with the patient because the somatic cell came from the patient. It takes nothing to obtain somatic cells from a patient, right? Just a swab of the mouth, for example, has somatic cells live. And the eggs, yeah, the egg is always the issue, is obtaining human eggs, of course, that would need to be nucleated and all that. But now the in vitro industry, again, is providing for nuclear eggs, right? Because for human eggs, because they have managed to cryopreserve human eggs. That was a big challenge, to cryopreserve a human egg in contrast to human sperm, because the egg, in contrast to sperm, has much more cytoplasm, which is mostly water, and the freezing and thawing of that water was a challenge up until more recently. Now they've managed to tweak the technique and go through so many human eggs, destroy so many human eggs, that they finally tweaked it to get human eggs to be cryopreserved, all right? And it was mostly the issue of the water content in the cytoplasm. Anyway, that was the whole purpose for this background, for this protocol, to eventually generate ICMs that were of the same origin, the same genome as the patient. So you can see, I mean, specialized medicine, right? It takes personalized medicine to a whole other level, and then generate the cell lines that were needed, so on and so forth, what I talked about earlier. Okay, so you see, mostly the motivation, the big picture motivation behind all this was for therapeutic cloning of humans. Now, before going into reproductive, let me finish the argument here, because I mentioned several times that Dolly was euthanized. What happened? As Dolly grew and bleeped, what is it called? Is it called bleeping? What the sheep do, the bah. As Dolly grew and developed and so forth, she looked normal at birth, she behaved normally. In fact, the actual full proof of principle was what? Just like with in vitro fertilization, and Louise Maynard, Louise Brown, right? They had to wait till when? For Louise Brown to declare the full proof of principle that in vitro was that was safe, that could generate a normal human being. They had to wait for Louise Brown to procreate, to have children. In other words, the full principle was if she was fertile, okay? Because let's say that in vitro works, but it generates infertile individuals for whatever reason, then it's not acceptable, right? Yeah, well, we can get you a baby, but that baby is going to be sterile. So that's why they had to wait really for decades. And that's why Edwards and Steptoe, the two British doctors who invented the in vitro back in the 80s, didn't get the Nobel Prize until a few years ago, precisely because Louise Brown had not had children, and they're not going to tell her, okay, go and reproduce for us so that we can prove the principle. Okay, we're dealing with human beings. So anyway, they had to mate Dolly, and for mating Dolly, they had to wait until Dolly grew up to sexual maturity. In other words, start ovulating and all that. And they did, and that took I don't know how many years. And they mated her, and she had baby sheep, and the baby sheep looked normal and grew up normal and so forth. But then, as Dolly reached maturity, actually, they realized that she reached maturity actually faster, chronologically, than the average sheep. And also that she started developing diseases that are associated with senility, like arthritis, and I don't know how they measured some kind of senility of the brain and so forth. But she was developing diseases that are, or illnesses that are of the elderly prematurely. She, by chronology of the sheep, should have been in the prime of her time, and she wasn't. She was declining. And so for humane reasons, they euthanized her. But at the same time, they realized that this was happening with mammals, because by that point, I don't want to say it became fashionable, but many other mammals started to be cloned. Once the technique was proven valid, that it worked, then other mammals were being cloned, beginning in two big industries. So one industry was the animal husbandry, cattle industry. Let's say you have a cow that for whatever reason produces a lot of milk. Well, you want to clone those cells, right? Because then you want to keep that going in other cows. So if we can take that one cow who produces a lot of high quality milk, all right, obviously her genome for milk is top of the line. And so if we clone that cow now and make 100 of those cows, and their output, the milk output is equivalent, well, that's fantastic for the industry, right? For the milk industry and so on. You can make the same argument for any part of the body that is commerciable for food and whatnot. Okay, so the animal husbandry is the one that ran with this, and they started cloning other cattle and animals that are using that. And the other big industry, of course, is going to be the pet industry, right? The pet industry, because as we fall in love with our favorite pet, and the pet is getting old and eventually she's going to die, we could clone the pet and keep it going. Or God forbid, if the pet had an accident, tragically, we can still say there are literally millions of somatic cells that are available even from that cadaver, right, that carcass, and clone him or her and keep it going. It's expensive, but it's part of, and it's something that the pet industry now offers now. It's certainly boutique, but it's offered, and a number of people actually have the money and the desire to go for it. So these are the two industries that took off with mammalian cloning, and now it's been done in many mammals. And let me just finish this thought, please. So what was happening, what they've noticed with these expanding the number of mammals that are being cloned, two things are coming to the fore. One is that they tend to age prematurely, invariably, and then develop these illnesses. And so for humane reasons, they're euthanized, okay, because they were going to be in pain, especially with something like arthritis. They're going to be in pain, and so they're going to be euthanized, you know. But the other thing that also came to light is that it's taken, so one thing that people didn't know, the general public, I don't think it was publicized in the first, when Dolly was generated, is that it took about 250 trials for this to work, all right? When I first described it, and then when I first read it, I thought it was just, oh, okay, 1, 2, 3 trials, and we're good to go. No, it took about, I think it was actually the 258th trial, but it took about 250 trials for this to finally work, all right? So you can see that when we translate that into the human, and this number of the 200s, if I remember, is about the average for the mammals that they were doing at the site. I was following, for example, another one that was cloned fairly fast was a cat, and it was called Copycat. You know, Copycat was the first cloned cat, and so on and so forth. They were coming up with these funny names. But basically, the number of trials was about the same, about 250, because it's a mammalian cloning model. You know, we could expect the average to be about the same, about 200 plus. Now translate that into the human. It means that for every successful human clone, 200 plus didn't make it, were sacrificed or died at some stage, you know, and these deaths are not natural, because there's certainly nothing natural about this technique. It's totally artificial, and therefore these are procured deaths of these human beings, right? And so that's a big ethical issue with regards to human cloning that people normally don't know, is the number of trials that it takes to get one successful. All right, so those are the two huge issues there, just on the science of it. Okay, sorry. So, Dr. Farid, was it you had a question or someone else?
Oh, it was me.
Oh,
I was.
Brother Stephen, yes.
The reason for the clones being, I guess, not advanced in age, but is that related to their telomere length because of the?
Exactly. Wow, you got it. Do you have a doctorate in genetics too? Yes. Excellent. Yes, the tips of so we know the tips of the chromosomes, right, are the telomeres, and every time there is mitosis or meiosis, the telomeres begin to shorten and shorten, and there is a repair mechanism that does that, re-adds some of the telomeres back, but it's never 100%. So if it's 99.9%, we always lose a very small percentage with every successive mitosis or meiosis of the tips of the chromosomes, and that has been associated somehow with the aging process. Exactly. Okay, so that's it, exactly, and that was happening to Dolly. Somehow, her cells were reproducing faster, were metabolizing or not metabolizing, were reproducing faster, and so she was aging faster than normal. Okay, and that's something that you see in nature. Nature doesn't forgive, right? This is such a totally artificial mechanism of generating a new individual of whatever species, and that nature takes exception to it. And there's certainly, it's very crude, all right? There's certainly very refined issues here. You can see already that, well, the somatic nucleus, think about it for a moment, the somatic nucleus is not pristine. The somatic nucleus has been exposed to this world for a number of years or months or even just days, whatever, and it's not new. So that somatic nucleus has already undergone trillions of cell divisions from the time that it started as a zygote and went forward into full differentiation. How many mitosis have occurred there? I don't know, but I'm gonna really, I mean, it's been millions, if not trillions of mitosis by the time that somatic nucleus is available, all right, for the transfer. And so you see, it's not pristine. And it carries its consequences, its negative consequences, which was only seen after post facto, was not expected, but in hindsight makes sense, because this is really a very crude technology for generating a zygote. All right. Okay, last thing I want to say about Dolly, tongue-in-cheek, just because I mentioned from the sublime to ridiculous, right? That this was, at some point, it was even it was getting comical, but not so much when you think of it. Why is Dolly called Dolly? Why was this why was the first clone mammal called Dolly?
Dolly Parton, the singer.
Yes, indeed. Yes, indeed. This is for posterity. Okay, think about this. This is in the history of humanity for posterity. In fact, that's the reason why I mentioned this is taxidermed. That's why Dolly was taxidermed, and she's now in a museum in the Rosling Institute, I believe, in a case, in a glass case, there to be preserved. Like other animals that we see in museums, right? And it is because of Dolly Parton. Okay, it sounds comical at first, but it also has, in my opinion, has ethical implications. So we know that the Dolly is an identical twin to this who donated the nuclear, the somatic nucleus, right? And we know from Dr. Wilmot that the somatic nucleus came from the udder of this sheep, of this donor, okay? And in Wilmot's own words, and you can look it up, in Wilmot's own words, he used the name Dolly because he says this is in honor of the prodigious mammary glands of Dolly Parton. That's why, just tongue in cheek, he ascribed to himself the privilege of taking her name for this because of the origin of the somatic cell. Yep, and so that to me completes the circle, right? You can make of it what you will. I happen to think that Dolly Parton is a very respectful actress, have high respect and regard for her, you know, and I'm sure she didn't take lightly to that, but that's what it is, okay? And it's there, again, you can look it up online. That's what motivated Wilmot to name the first clone mammal, Dolly. Okay, folks, so sometimes we just get a little too brazen, right? Questions about reproductive cloning? Sorry, I should label this therapeutic cloning. I just noticed. Well, technically it's reproductive, but the motivation behind it was therapeutic. All right, so reproductive cloning, well, basically it's a rehash of what I've just been talking about. But with this diagram, I just want to point out that the number of parents that the clone will have, all right, so you have the donor egg, who is, which is enucleated. And now we have a donor cytoplasmic ovum, which will be partnered with a somatic cell, either from the same woman or from a donor woman. All right, so there's a possibility of a second mother there. and even mother in quotations. Now we do the fusion and we have a zygote. The zygote has, in this case, has two mothers, right? There's no father inside because there's no sperm. And then that zygote is incubated to the blastocyst stage, implanted. In principle, that could be implanted into a third woman, the surrogate, for gestation until birth. So at the end of the day, this baby, who is obviously going to be a girl, can have three mothers and no father. And at least one of those mothers is her twin sister. In other words, the one who donated the somatic cell, right? Simultaneously mother and twin sister, but with years, decades of age difference. And in all this, has anyone consulted with this baby that she wanted to have? Three mothers, no father, biologically speaking. I'm not talking about adoption. You add the adoption layer to this, you can add one more mother and one more father, okay? And one of those mothers is also her identical twin simultaneously. And what about, it doesn't take much rocket science to think when this child grows up and this girl starts asking, well, who is my mommy? Okay, and why is this mommy my twin sister? Okay, so there are obvious ethical implications here for why reproductive cloning should not be done. And yet, really, none of these ethical reasons were used for banning reproductive cloning, which there is a ban on reproductive cloning, at least with federal money. That's so. How do you do, how do you do these bans? These bans are on federal money, right? Because it's public money. So that's why NIH or NSF can put bans on certain research, because it's public money, and they have to poll the people to see if it's a go or no go. And people overwhelmingly are against cloning, reproductive cloning. They don't know the various, most people don't really know the distinction and so forth, but the scientific community also came out big time against reproductive cloning. I'm thinking of that initiative that was done in California precisely with for generating that institute, the California Stem Cell Research Institute, because they are also involved in the cloning process. There is a federal ban on human reproductive cloning, but the main reason, many scientists, say an overwhelming number of scientists in the US and other parts of the world are against this, but the reason is precisely the senescence issue, the premature senility of Dolly and the other mammals, okay? That's the reason. In other words, that it's a grave injustice to generate a human being who is going to age prematurely. And it's true, it's a grave injustice to do that, all right? But that's their main reason for opposing reproductive cloning. None of this. So you can see that basically it comes out of a utilitarian camp, the utilitarian mentality that the end itself, not the means, these are the means, but the end itself is not justifiable, because one way or another, it creates a defective child, a child who ages prematurely, and now we know. So to do this knowingly is not ethical. And it's true, it's not ethical, because the product, if you will, the child is going to be defective, is going to age on average, based on what we know now, is going to age prematurely, you see? So there is a ban for reproductive cloning at the federal level in the US for that reason, but not because of the principle of the manipulation that is going on through here, okay? Finally, well, this is a rehash. Maybe I should reverse these slides, but basically, this is just a rehash of the therapeutic cloning, right? It's manufacturing the blastocysts for obtaining the stem cells. Let me just magnify this a little bit. Hope you can see it there. The patient, and it would be autologous. So here's the patient donating somatic cell. And if the patient is female, maybe she can donate the egg too. But if the patient is a male, then those somatic cells are inserted into enucleated donor oocytes, donor eggs, yes, obtained from in vitro clinics, right? And here's the embryo after incubation and all that to the blastocyst stage. The ICM then is disaggregated for the embryonic stem cells. And in principle, or theoretically, all these cell lines may be able to be developed from those embryonic stem cells. And at least that would be immunocompatible with the patient from where the somatic cell came. But we know that this is not happening. And so those trials are not going on either right now. Thanks be to God. Bottom line, neither therapeutic nor reproductive cloning are working. But then, never a dull moment. It was, I think it was last year that some scientist in China claimed that he had cloned a human for the first time. Now, it's really hard to tell because federal funds are not available, which is the big money, but that doesn't mean that state or private funds aren't available, right? And in fact, they are. Like an example I mentioned several times, in California, state money is available for human embryonic stem cell research. And and with private money, well, who knows, right? But Dr. Hay, I don't know if you heard, it was in the news last year, H.E. Cloning. That was a huge, there it is, you can read up on it, but basically he claimed he had done human cloning and he did it with 2 baby girls. Of course, he didn't say it until the baby girls were born. Their names are Lulu and Nana. I don't know if you've heard those names. It was in the news, Lulu and Nana. And supposedly he engineered the somatic nucleus so that they would be immune to HIV, all right? Immune to HIV. And then he did a whole procedure in secret in a lab that he was working in China somewhere. And you can imagine, I mean, all kinds of ethical issues, right? These, according to him, now to this day, he has not presented the scientific evidence for all this. He just blurted it out at some kind of a scientific meeting that was going on. Seemingly he had some poster on it, but then he disappeared, and even the Chinese government was after him because he broke all kinds of rules that you can see here by doing this. And just on the face of it, how do you test? You know, are you going to infect Lulu and Nana with HIV to see if they're really immune or not? Really, are you going to do that, right? So you see, it was just totally bizarre when we don't take care of the ethics. It's horrible. And supposedly, I mean, at least by his claim, these 2 girls exist, they're growing, and they're somewhere on this world, okay? So that's the challenge when we don't really consider the ethics behind this stuff. And the pressure, of course, the scientific pressure is so strong to publish or perish, right? That he thought he was going to be the first one to do human cloning. And he was going to do it for the good, right? In fact, I think he actually used CRISPR for inserting the genes immune to HIV and so forth. Mm-hmm. So that's what can happen when we don't take care of the ethics before we do the science. Okay. Okay, folks, it's 12:32. So let me stop. Questions, comments?
Yeah, Father, this is Frankel here.
Yes, Father Franco.
Yeah, you just said about when we don't take care about our ethics before science. So, well, not concretely, it is that concretely. So what we say with this true form of ethically of the form of cloning, if like we receive a call last week, we will receive some calls too, still receiving this call. So what way we are going to help people understand that even this kind of the cloning in their understanding.
Exactly. And thank you for the question. And that's also why I sent you the PowerPoint in addition to the video and so forth, because going forward, maybe you didn't know much about cloning 3 hours ago, and I'm not saying you, Father Franco, but the class in general, all right? But now you do. I'm sure that for a number of you, you know a little bit more about cloning now than you knew 3 hours ago. And for some of you, you know a lot more than you knew 3 hours ago. And so that's knowledge now is power, right? And knowledge is for illumination. Now you can talk to others about human cloning, and you can use the PowerPoints. You may not have to go in this level of detail. It's always my problem of, I never know how much detail really the student is going to need, because the cohorts are made up of such varied backgrounds. But there it is, just in case. I try to put it out and give you the references so that now I hope that you feel competent in talking about stem cell research and cloning to others, right? And you can see that the first thing is making all those distinctions, bringing it down to the human level, and then what's involved in the technique, so that you can speak with competence without really having the terminal degrees, because this is just one area of bioethics. It's pretty specific. But yeah, you may be getting these questions from people at some point. Or you may be, for those of you who are aspiring to medical school and the medical profession, certainly, I would hope that this is covered in medical school, but invariably, my intuition is that it's going to be covered from the utilitarian perspective, okay? So at least you have an alternative here, if it's offending your ethical sensitivities, that you have something to defend your value system with. Mm-hmm. All right. I know, Franco, Father Franco, did I do justice to your question?
Yes, thank you.
Mm-hmm, sure. Okay, so basically resources, right? It's just to give you a toolkit for resources to use.
Father, this is Farid, and another point is that in addition of killing the embryo, this is one, and in addition of that, you can try to create a superior race, people that is.
Oh yes, well, exactly. Obviously,
this is something that it can come at any moment with these procedures.
Right, and you bring up a very important issue, which is the can and the may, right? Always going back to that in bioethics. Absolutely, and in a sense, this is nothing new. Obviously, all these techniques have the potential for eugenics, right? Eugenics, And we know about positive eugenics and negative eugenics, and it's less than a century ago that it led to a Second World War. Horrible, horrible, to the tunes of millions and millions of people being killed or maimed and just entire lives destroyed. So this certainly can lead into eugenics. And it's not new. You can look up a movie called The Boys from Brazil, way back, The Boys from Brazil, which was precisely cloning Hitler. Yes, in Brazil. Okay. After the Second World War, we laugh about it, but you know, any one of ourselves can do it. Hmm?
No, I saw the movie, 1976 or something.
Is it really? Wow, look at that. Yeah, okay. Yeah, it goes back half a century, but it's there. And obviously, technology has not stopped in the past half century, right? If anything, it's gotten much more sophisticated. Again, I go back to CRISPR. It's amazing. The power that we have today in a lab under the microscope for impacting humanity for the good or for the evil. All right. And so we really need to take assessment because next generation will judge us for what we're doing. What we're doing. I'm not going to delay this any further, but I'll bring this topic back up again when we look at the lecture I'll give on in the healthcare bioethics on vaccination, which, by the way, for the group on the 510 course, right? That will just be a lecture that I'll send you the video for it and the PowerPoint and all that, because there's no time to cover it live in the program. For the Masters in Bioethics at STU, we have an entire course on healthcare, which expands, and we'll look at it in detail. But for the 510 course, I'll just send you the materials there. You can look it up on your own. You can watch it on your own. Okay. All right. So once again, thank you very much for paying attention on another heavy topic. Hopefully it has been useful to you and to the people you encounter going forward. With that, I'm closing and thanking you. We should be back live. Hopefully I'll be back at STU live next Saturday, okay? And we'll be starting.
Thank you, Father.
Okay, we'll start the human life on the end of life issues next Saturday. Now, bye-bye, everyone. Close here. Where is it? Is it here somewhere? Here we go. Thanks again. Bye.